Today's topic is the cells of the immune system. Under the cells of the immune system, there are two types of lineages, that is lymphoid lineage and myeloid lineage. These lymphoid lineage and myeloid lineage cells arise from a single cell called as hematopoietic stem cell. Okay, hematopoietic stem cell is a one kind of stem cell. Okay, so we all know what is stem cell. Stem cell has the ability to differentiate into number of different kinds of cells. It has the ability to differentiate into what different kind of cells. Okay, they can acquire a different morphology and characteristics, functions of different kinds of cells. Okay, so all the cells which come under the cells of the immune system arise from a single type of cell, that is hematopoietic stem cell. All the cells of the immune system arise from a process called as hematopoiesis. Okay, so hematopoiesis is the process from which the all cells of the immune system will arise. Okay, under the process of hematopoiesis, there are two types of categories of cells, that is lymphoid lineage cells and another is myeloid lineage cells. Under the lymphoid lineage cells, okay, these are also called as lymphocytes, okay, which is a part of adaptive immune system. Okay, there are basically two types of immune system, that is innate immune system and adaptive immune system. Okay, so in the adaptive immune system, which consist of lymphocytes, okay, lymphocytes, that is B lymphocyte, T lymphocytes, okay, which will see further. Okay, so lymphocytes consist of 20 to 40% of body's white blood cells and 99% of cells in the lymph. The lymphocytes can be broadly subdivided into three populations like B cells, T cells and natural killer cells on the basis of function and cell membrane components. So due to that, the presence of B cells and T cells, these lymphocytes are the components of adaptive immune system, because the B cells and T cells has the characteristics called as memory. Okay, so B cell and T cell has the one feature, that is memory. So means these cells has the ability to convert into the memory cells. Okay, memory cells, when they are converted into memory cells, when there is exposure of antigen. Okay, so when there is exposure of antigen, these cells get activated. Okay, and they will convert into effector cells and memory cells. Okay, so memory cells, what they do, they will keep the memory of antigen, and when the same antigen is encountered by the body, okay, the memory cells will get activated and they will perform the action against that antigen. Also, all these three cells also have another attributes like specificity, diversity and self and non-self recognition. Okay, means what? It can recognize the antigen is self or non-self. Also, it has diversity. Means what? It can kill as many as antigen as possible. Okay, specificity means what? It can all these three cells can kill a specific kind of antigen. B lymphocytes derive its later designation from its sites of maturity in the brusa of Fabricius in birds. Bone marrow is the site or major site of maturation in number of mammalian species. So the B cells acquire its name from the site of maturation. Means what? Where the B cells is going to mature. Means what? The B cell in birds is going to mature in the brusa of Fabricius. Okay, and more bone marrow is the site of maturation of B cells in the mammals. Okay, so B cells display a membrane bound antibody. Means what? B cells are the cells which produce the antibody in the body, okay, or in the human body. Okay, so mature B cells display membrane bound antibody. Means what? B cells when encounter the antigen, it will first get converted into the plasma cells. Then these plasma cells will produce or secrete the antibody, okay, which will be the membrane antibody, which can be the which will act as receptor on the B cell. Each of the mature B cell contains 1.5 into 10 raised to 5 molecules of antibody on the membrane of the single B cell. Okay, means what? Each and every B cell will have 1.5 into 10 raised to 5 molecules of antibody, means that number is 1.5 into 10 raised to 5. Okay, so each B cell will have this number, okay, identical, which will have the identical binding site for the antigen. Interaction between antigen and membrane bound antibody on mature new B cell, as well as interaction with T cells and macrophages, selectively induces activation and differentiation of B cell clones of corresponding specificity. Means that new B cell which is not encountered any kind of antigen, okay, means what? New B cell is a new type of cell, okay, means it will not get interacted with any kind of previously antigen, the which is not differentiated yet. When the B cell interacts with the antigen, it get differentiated. So new B cell means what? It is not interacted with any kind of antigen. When the B cell interacts with any kind of different type of antigens, it get differentiated into the new types of cells. Or when the B cell, that is new B cell, which is not encountered with the antigen, when it interacts with the new antigen or T cells and macrophages, it selectively induces activation and differentiation. In that process, the B cell divides repeatedly and differentiates over 4 to 5 days and generates a new population of cell, that is plasma cells and memory cells. But this all will happen when only the naive B cell will interact with new kind of antigen. Until and unless it doesn't interact with antigen, the naive B cell will not differentiate into the plasma cells and the memory cells. Plasma cells which have lower levels of membrane bound antibody than B cells synthesize and secrete the antibody. Means our main focus to kill the any kind of antigen is to produce antibody. Means the antibody, which is the molecule which will going to kill the antigen. So the B cells should be differentiated into plasma cells. The plasma cells are the cells which are going to produce or secrete the main antigen killing molecule called as antibody. So plasma cells circulate in the blood for 1 to 2 weeks. After that they will get die, and the new, if the body encounters any new kind of antigen, then the plasma cell will get again differentiated from the new B cell and the cycle will continue. T lymphocytes derive their name from their site of maturation in the thymus. Like B lymphocytes, these cells have membrane receptor for antigen. They are called as T cell receptors. Unlike membrane bound antibody on B cells, which membrane bound antibody on B cells, B cells have membrane bound antibody, okay, so they will recognize the antigen, any kind of free antigen. Same like that the T cell can't do. The TCRs does not recognize any kind of free antigen. Means what? The antigen must be complex with the MHC molecule, that is major histocompatibility complex. When the antigen is complex with the MHC molecule, that complex, that is antigen plus MHC molecule, will be recognized by the T cell receptors and eventually the T cells. But alone or the free antigen does not recognized by the T cell, which is the we can say the drawback of the T cell as compared to the B cell, because B cells alone can recognize, or the antibody molecule which is present on the B cell will can uh, recognize any kind of antigen which is free, but the T cells can't recognize the free antigen. It must be complex with the MHC molecule. So antigen plus MHC molecule, this complex or the whole complex will be recognized by the T cells. So to be recognized by the most T cells, the antigen to be recognized by the most T cell, this antigen must be displayed together with the MHC molecule on the surface of antigen presenting cells. Means antigen presenting cells like dendritic cells, B cells are the antigen presenting cells. Means will present to antigen to the T cells. Okay, so this antigen must be complex, first must be complex with the MHC molecule, and this antigen plus MHC molecule will be displayed on the surface of antigen presenting cells, and these antigen presenting cells will present this whole complex to the T cells. In that way the T cells only can recognize the antigen, but the T cells can't recognize the antigen which is free, or the T cells can't recognize the antigen as alone. T cells that express membrane bound glycoprotein molecule called as CD4 and CD8. Okay, so on the basis of the CD4 and CD8 glycoprotein, T cells can be designated as CD4 T cells and CD8 T cells. CD4 are restricted to recognize the antigen bound to the class 2 MHC molecule only. Means that those antigen which are bound to class 2 MHC molecule will be recognized by CD4 plus T cells, and whereas the T cells expressing CD8, a dimeric molecule glycoprotein, are restricted to recognize the antigen bound to class 1 MHC molecule. Means those antigen which will be bound to the class 1 MHC molecule will be recognized by the CD8 plus T cells. CD4 plus and CD8 plus T cells are again differentiated. Okay, so CD4 plus T cells generally functions as T helper cells and are class 2 MHC restricted. CD8 plus T cells generally functions as T cytotoxic cells and are class 1 restricted. So now we have seen earlier that when antigen encountered by any type of cell, then and there it will get activated. So T helper cells are activated by recognition of the antigen with class 2 MHC complex on the antigen presenting cells. Means what? The class 2 MHC complex plus the antigen presenting cells plus antigen, this whole thing or whole complex will be recognized by the T helper cells, or the whole complex will go towards the T helper cells, and in that way the T helper cells will get activated. So these T helper cells secrete various kinds of cytokines which will kill the antigen eventually, which play a central role in the activation of B cells also. Means that T helper cells will also activate the B cells also, but after the activation of T helper cells. Until the T helper cells has not activated, it will be not able to activate the B cell also. T cells, B cells and the T cells and other cells that participate in the immune response. Means what? The T helper cells are eventually activating the B cell, T cells and other type of immune system cells also. So there are two types of T helper cells, that is T helper 1 and T helper 2. T helper 1 response produces a cytokine profile that supports inflammation and activates mainly certain T cells and macrophages. So certain different kinds of T cells are also there, which will be activated by T helper cells, and some kinds of macrophages, okay, which will eventually lead to the deletion or deletion of the antigen, or killing of the antigen will be done with the help of different type of T cells and macrophages, which will be activated by T helper 1 cell. And the T helper 2 cell response mainly activates B cells and the immune response that depend on the antibody. So antibody mediated immune response will be activated by the T helper 2 immune response, or T helper 2 response, or T helper 2 type of cell. So the different kinds of cells which are present in the immune system will be activated by T helper 1 cell, and the antibody generated kind of immune response. Under the cells of immune system, the second type of cell is myeloid cells or myeloid lineage cells, under which the myeloid cells are also called as granulocytes because their cells have granules in their cytoplasm. They are classified as neutrophils, basophils, eosinophils, mast cells, dendritic cells, and follicular dendritic cells. Under which, first, the neutrophils. Neutrophils constitutes about 50 to 70% of total white blood cell content. It has multi-lobe nucleus. The cytoplasm of neutrophil is granulated, and the neutrophils are stained by acidic or basic dyes. Then the next cell is eosinophils. It constitutes about 1 to 3% of total white blood cell content. So it has bilobed nucleus and granulated cytoplasm, and the eosinophil stains by the acidic dyes. Next cell is basophils, which constitutes about less than 1% of total white blood cell content, and it has a lobed nucleus and heavily granulated cytoplasm, and basophil stains with the basic dyes. So due to the basophil stains with the basic dyes, the name of the basophils is basophils. So neutrophils is the first cell, under which neutrophils are produced by the process of hematopoiesis in the bone marrow. They are released into the peripheral blood, and they circulate there for 7 to 10 hours before reaching to the tissue species, where their lifespan is only few days. In response to many types of infections in the body, when there is any type of infection or inflammation which occurs in the body, neutrophils are the first cells which are recruited at the site of infection. Okay, so neutrophils are the cells which are recruited at the site of infection firstly, then the other type of cells when there is any kind of infection or inflammation which occurs in the body, so that the infection can be eliminated by the neutrophils. So the immune system of our body releases number of neutrophils at that site of infection to remove or to eliminate that infection, and the result is that the increase in the neutrophil count in the body. So total number of neutrophils which are present in the body is increased. That is, it is called as abnormal condition, and that condition is called as leukocytosis, which can be used as medically as an indication of infection. Movement of circulating neutrophils into tissue spaces is called extravasation, takes place by several steps. Like, first step in the extravasation is The cell first adhere to the vascular endothelium, then penetrates the gap between adjacent endothelial cells lining the vessel wall, and finally penetrates the vascular basement membrane, moving out into the tissue spaces. Means what? To enter into the tissue spaces, the circulating neutrophils will perform the function, or will perform the extra kind of step that is called as extravasation. Okay, so when the number of substances generated in the inflammatory reaction serves as chemotactic factors that promotes the accumulation of neutrophils at the site of inflammatory site, means where the infection is occurring or inflammation is occurring there. At that site, some of the chemotactic factors or other molecules will get accumulated. Chemotactic factors means what? It will attract the uh, chemical like substances. On the other hand, due to the accumulation of chemotactic factor, it will help to promote the accumulation of neutrophils at the site of inflammation or the site of infection. Neutrophils also generate some antibacterial or antimicrobial substances to kill the infection or to remove the inflammation from the body. So neutrophils uses both oxygen dependent and oxygen independent pathways to generate the antimicrobial substances. The next type of cell is eosinophils. Eosinophils, like neutrophils, are also motile phagocytes that can migrate from blood into the tissue spaces. Their phagocytic role is significantly less important than neutrophils, means they are less important than neutrophils. Neutrophils play a very major role in the elimination of the infection or removal of inflammation from the body than the eosinophils. They are thought that they play a role in the defense against the parasitic organisms. The next cell type is basophils. Basophils, unlike neutrophils and eosinophils, they are non-phagocytic granulocytes. They function by releasing pharmacologically active substances from their cytoplasmic granules. Means their cytoplasm contains pharmacologically active substances which will release some kind of chemicals which will remove the infection or inflammation from the body. These substances play a major role in certain allergic responses. So the basophils are generally play a major role when there is allergic reaction faced by the body. Next cell type is mast cells. Mast cells precursors, which are formed in the bone marrow by the process called as hematopoiesis, which are released in the blood as undifferentiated cells. They do not differentiate in the blood cells, or they are released in the blood as the undifferentiated cells. They will differentiate in the tissue spaces when they will reach in the tissue spaces, until and unless they will act as, or they will remain as, undifferentiated cells. Mast cells can be found in wide variety of tissues, including skin, connective tissue of various organs, and mucosal epithelial tissues of respiratory, and also the digestive tracts. Like basophils cytoplasm, the mast cell cytoplasm also have the cytoplasmic granules which contain histamine and other pharmacologically active substances which are used in the allergic reactions or to remove the allergic reactions. Mast cells with the basophils togetherly with the basophils plays an important role in the development of allergies, or to when the allergy occur in our body. Okay,